All right, well, I'd like to welcome everyone to this morning's Polyploid webinar. Today we have a couple of great graduate student speakers who are going to tell us about polyploidy at very different scales and sort of at the different spectrums of the field of study. First up is Thomas Anneberg from Syracuse University. Thomas has a BS in biology from the University of Kansas, and he's currently in Carrie Seagrave's lab at Syracuse, where he's a fifth-year PhD student. He's broadly interested in plant nutritional ecology, species interactions, and fungi. Thomas is going to tell us a little bit about his work today in Heuchera. In particular here, Neopolyploid Heuchera cylindrica are more nutrient limited. And I look forward to hearing about that. And so I'm going to let Thomas take it away.
All right, thanks, Mike. Today I'm going to be talking about how Neopolyploidy and Heuchera can cause nutrient limitation to be a little worse. So before I get into that, I just want to highlight how speciose polyploidy is across the plant kingdom. So not only is it really speciose, and we have lots of examples where polyploidy occurs, but I have some photos here where there's examples of interspecific variation in ploidy. So there's multiple cytotypes within a single species. So not only is it really common, but these polyploids are forming all the time, repeatedly through time, I should say. So a good example of this is in the Tragopogon triangle, where the diploid parents are giving rise to neotetraploid or first generation tetraploids repeatedly. So just to kind of illustrate that example with another cartoon, we can imagine a diploid clover species giving rise to very many neopolyploids. So with Neopolyploidy, there's some demographic challenges. So the Neopolyploids are immediately competing with their diploid parents for niche space. But also I've kind of illustrated the Neopolyploids as being a little bit bigger. So that's one of the strong morphological correlates of polyploidy. We'll talk about that more in a minute, but with these demographic and what I will call physiological challenges of polyploidy, we expect many of these Neopolyploids to go extinct shortly after they arise. So a really big focus in Neopolyploid ecology today, or polyploid ecology more broadly, is understanding why Neopolyploids, the ones that ultimately become established, do so. How do they survive and persist through evolutionary time? Just to kind of go back to those morphological correlates, I'm using Arabidopsis as an example here, but there's many other examples of this phenomenon where polyploidy can increase size-related traits. So you can see on the left here, the flowers and the trichomes just get bigger with ploidy level. And similarly, leaf size and other traits just get really big with ploidy. So I think that that's a really big nutritional challenge for the polyploid to overcome in terms of being able to facilitate growth in the same environment that they're born from, which is the diploid niche space. So I think the idea that neopolyploids are immediately more nutrient limited than their diploid parent isn't necessarily a newfangled idea. There's been some recent literature from long-term fertilization studies in grasslands. This study on the left that I'm going to show you first, they supply different amounts of phosphorus and calcium to plots. And where you see an increase in biomass is where they supplied more nutrients. And another study from 2016, which I really admire, the authors supplied both nitrogen and phosphorus to field plots, and they saw with increasing nutrient supply, these gray triangles on the figure here, the established polyploids in the field grew more, or responded more positively to nutrient enrichment than the co-occurring diploid species. So each triangle and each dot represents a species that had become established at those field sites. So the polyploids are responding more positively to nutrient enrichment. So that to me tells me that polyploids are a little more resource limited than co-occurring diploids. And I wanted to go upstream of the establishment process. So before establishment had occurred and within lineage, how does neopolyploidy affect nutrient limitation? And a good example to kind of show you how neopolyploidy might, if you look at a neopolyploid, you might get a disparate response if you looked at an established polyploid instead. Brian Husband in 2016 synthesized a number of neopolyploids from Chimera angustifolium or fireweed. And you can see on the left, the diploid parents and the established polyploids on the right are kind of on the polar extremes of the morphological trait variation that he saw. The neopolyploids have an intermediate phenotype. So if you compare the polyploids that it established to the neopolyploids that had been de novo synthesized, you see that this post-whole genome duplication evolutionary forces have ostensibly been pretty strong in forming the trait responses for the polyploids that were looked at today. So I think that kind of highlights the importance of looking at neopolyploids to really understand the direct effect of whole genome duplication. So that leads me to my big question for today. So this is going to comprise the majority of the rest of my talk. How does neopolyploidy affect plant responses to nutrient supply? And then at the end, I'm going to also pay a little bit of credence to the question, are the effects of neopolyploidy consistent across multiple origins? So I used Heuchera cylindrica to address my questions. This is a small perennial herb that grows in the Pacific Northwest. It's been used extensively to study plant ecological interactions as a consequence of polyploidy, but it also forms the storage organ, being a perennial plant, that can allow us to kind of at least hint at the idea that not only is there nitrogen and phosphorus limitation playing out in the species, but we can also look at the interface of carbon limitation too. So using this species that stores carbon for next year, that's a really valuable trait for us to assess how apt the plants are at acquiring that resource too. So I went up to the Pacific Northwest of the United States again, so mostly Washington, but Idaho as well. I collected 12 maternal diploid lineages from these corresponding 4 diploid sites, these filled circles in Washington and Idaho. And from those 12 maternal diploid lineages, I synthesized a corresponding neopolyploid. And then we also collected 4 established tetraploid lines. So those open circles in the panhandle of Idaho also constitute our 2 sites that we used. So I did include treated diploids with our polyploidizing agent that we made our neopolyploids with. So I'll say failed induction diploids. We put those into our model and statistically grouped them in and looked for differences between the diploid state and the untreated diploids versus the treated diploids. We saw no statistical difference, so I didn't want to complicate the matter by including a 4th group of plant lineages, but we did test that and there wasn't really an effect of nitrous oxide gas, which is what we use to make our neopolyploids. This is just kind of an interesting aside because a lot of people use colchicine, but we use pressurized nitrous oxide gas, which is reappropriating a method from the early 1950s, just showing you what the apparatus looks like. We've had really good luck with Heuchera cylindrica and a couple other plant species in making neopolyploids. So I'm happy to talk about that during today's Hangout session after the webinar today, but I don't have a whole lot of time to go into that right now. Our nutrient treatments for the control value of nitrogen and phosphorus, we based that on springtime field conditions from the field sites. So we looked at the available nitrates and phosphates available from the field, and we slightly skewed the amount of phosphorus up just so that we got a molar ratio of nitrogen and phosphorus of 16, which is often considered optimally co-limiting for growth of plants. And we either quartered or quadrupled the amount of nitrogen and phosphorus to reach our low, control, and high nutrient treatments. So for the biomass traits that we measured, we of course looked at the total biomass production of plants. And then from the total biomass produced for the plants, we looked at the relative allocation of that biomass to different structures. So we looked at the above ground mass of plants, allocation to rhizome mass or carbon storage, and then we also looked at the fine root mass of plants. We were interested also in photosynthetic parameters, so we looked at a few leaf traits. So we measured specific leaf area, which is the ratio of leaf area to leaf dry mass, and increasing leaf area means that the leaf is stretched more thinly over a bigger area, so that's viewed as a more acquisitive leaf for carbon. For that reason. And then we also looked at the dry matter content of leaves, which is the ratio of leaf dry mass to leaf fresh mass. And that scales positively with leaf longevity. So having a high dry matter content means you have a longer lived leaf. So you might get more of a return on investment for carbon gain. And then it also has an inverse relationship with relative growth rate. So it means you're a slower growing plant most often if you have a high dry matter content. And then we also looked at leaf chlorophyll, or the concentration of chlorophyll in leaves, just as a measure of photosynthetic efficiency. We did go below ground too, and we measured the average fine root diameter of plants. So I view that trait as meaning if you have a really fine root, that means you're more able to probe a finer volume of soil more efficiently for limiting nitrates, phosphates, and other mineral nutrients. And then we looked at the concentration of nitrogen and phosphorus in those root tissues, the acquisitive roots specifically. And that's because that's where plants have the highest homeostatic control over their nutrient concentrations. So if we did see a higher construction cost of building a plant tissue, if you're a diploid or neopolyploid, this is where we'd most likely see it reveal itself because there's the least amount of plasticity in the plant here. So just to give you a moment with the first set of results, I'm going to have a lot of these reaction norm plots. So I'm just going to take a moment to explain. On the x-axis, we have our low, control, and high nutrient treatments respectively. The lime green lines represent the neotetraploids that we synthesized. Their diploid parents' reaction norms are the gold bars, or the gold lines, I should say. And then we have the established tetraploids that we incorporated in our study as the dark green bars. So the first thing you probably notice is that with increasing resource supply, the neotetraploids didn't grow as much as their diploid parents. And at the low nutrient supply, we see that there's no statistical difference between diploids, established tetraploids, and neotetraploids. So with increasing resource supply, the neotetraploids just grew less. And the relative allocation to different biomass structures. So this is a stack bar graph of the 3 biomass structures we looked at. We looked at above ground mass, vine roots, and rhizome. Excuse me. And then we have 2 panels. We have our low nutrients on the left and our high nutrients on the right. I took out the control treatment because the low and the control nutrient treatment elicited the plants to have a similar allocation strategy. So the low and control looked about the same. But on the right, if you look at the high nutrients, the neotetraploids in the middle didn't differ from their diploid parents on the left and their established tetraploids on the right and their allocation strategy. But with low nutrients, we see that neotetraploids we're allocating more of their biomass above ground compared to below ground into storage. So that was like the first hint of evidence that these neotetraploids were a little more carbon limited. So even at low resource supply below ground, the plants were still allocating more biomass above ground, and that was to the detriment of storage. And you can kind of see that playing out more clearly, just looking at the above ground biomass allocation strategy reaction norm, where the neotetraploids in lime green here were forming more biomass above ground instead of rhizome, particularly at low nutrient supply. So that's pretty compelling evidence to me, at least, that the neotetraploids were more carbon limited. And we see a similar pattern emerging from our leaf trait data, where the neotetraploids just had less acquisitive leaves. So if you remember what I said earlier, with increasing specific leaf area means you've got a thinner, bigger area leaf. So the neotetraploids, it appears that they had squatter leaves that were more fleshy because increasing resource supply, we saw no appreciable increase in specific leaf area. And so I wanted to just decompose specific leaf area into its 2 constituent parts to kind of show you how that pattern explicitly played out. So with increasing resource supply, the neotetraploid leaves did in fact get bigger in area, but that increase in area came at a really big cost. And it was more costly in terms of the biomass investment to make that bigger area leaf. So for what it's worth, the neotetraploids were trying to make bigger leaves. It's just that the cost of producing that was so high that specific leaf area at the growth scale was lower than their diploid parents. So just to kind of show you what the plants look like, I've got the diploid parent on the left here with these thin, small area leaves, and then the neotetraploid progeny in the middle. There's a pretty stark difference. I mean, clearly there's fewer leaves. The leaves are bigger in area, and I don't know how well you can see it, but the leaves were fleshier, as I said. So and the tetraploids that were established that we had from the field, those tetraploids, they had bigger area leaves. It's just that they were a little more thin. So it appears that there's been some selection against having a fleshier leaf. You can kind of see that in the dry matter content too. So this is the ratio of leaf dry matter content divided by leaf fresh mass. So this tells us how much structural carbon was in a saturated leaf. And we can see that the neotetraploids by and far had less structural carbon in a saturated leaf than diploids and established tetraploids. So in a perennial plant, this might be pretty disadvantageous because There's a positive relationship with leaf longevity. So if the neotetraploids have less long-lived leaves, then their diploid parents were perennial. That means that the return on investment for total carbon gain over a lifetime of a leaf might be lower. But we didn't directly measure photosynthesis traits like maximal photosynthetic rates of the leaves. So I can't necessarily say that directly, but it just appears that we've really got some less efficient leaves going on with Neopolyploidy. And as an interesting aside, the inverse of dry matter content is leaf water and gas content. So these neotetraploid leaves had more sugar and phloem tissue flowing around in the leaves. So that might play out pretty negatively for plant herbivore interactions. But I just wanted to make that quick aside because that was something interesting that popped out in the data to me. Below ground, we also saw that the neotetraploid roots were thicker than their diploid parents. So that was really cool to see. And the established tetraploids actually had an intermediate phenotype between the neotetraploid state and the diploid state. So it looks like there's also been a little bit of selection, maybe drift happening against having a thicker root. You can really appreciate the difference just looking at them, how much stubbier the roots are for the neotetraploids and their diploid parents. So having a thicker root means you're probably less able to get at those like really fine particulate spaces in the soil horizon for those limiting mineral nutrients. So the neotetraploids had less efficient structures above ground and below ground, it appears to me. And not only were the roots thicker, but at some nutrient treatments, the neotetraploid roots were more costly to build for nitrogen and phosphorus concentrations. So we've got root nitrogen and root phosphorus concentrations as a percentage in both of these figures. You can see with increase in resource supply, there was actually an increase in plasticity for the construction cost of a root. So high nutrient treatments, there was more nitrates and phosphates flowing into those roots to build them. So you've got a less efficient root being built because it's taking a lot more nutrients to build them. Having a higher root concentration might also mean that it's growing quicker just because of active division going on. Same with phosphates, but I don't necessarily have data to support that because we looked at all the acquisitive roots, whether it was meristematic or not. So just to kind of drive that all home, by and large, neotetraploidy caused the plants to be a little less efficient at using their resources and allocating them below ground. So I don't have any like whole plant pictures to show you with above and below ground structures, but if you can imagine, these neotetraploids, they're allocating a lot more of their biomass above ground than below ground, particularly into storage. So I see that as a pretty maladaptive consequence of neotetraploidy, at least in high resource environments. So I really do think it's at these low resource environments where there's not much difference, at least in total biomass production, where we might see these neotetraploids become established ultimately. But at these high nutrient treatment levels, we do see that the neotetraploids are a little more nutrient limited in the sense of carbon, nitrogen, and phosphorus than their diploid parents. So getting into my second question just for a moment, when I incorporated population of origin as a main effect, so the interaction between plant cytotype, nutrient treatment, and population of origin, on the left, I have the 5 traits where it didn't matter for like the population of origin on the trait response of diploids and neopolyploids. But on the right, we have 4 traits where we saw a significant effect of population of origin on the trait responses of plants. And that was the allocation to above ground mass and rhizome storage, specifically fairy leaf chlorophyll. So I only have time to really show you a couple of examples or a couple of those traits. So I'll look at the most stark example to me was leaf chlorophyll, where I've got my 4 sites, my 4 diploid sites that we collected our seed stocks from. On the top, you can see that Cheney, Washington, Blue Mountain, Washington, and Santa, Idaho, there was no effect of population on leaf chlorophyll content. There was also no effect of cytotype on leaf chlorophyll content in those leaves. But in Palouse, Washington, there was a significant effect of population of origin and cytotype on leaf chlorophyll concentration. So that was something that really jumped out to me and I thought was really cool. that an independent origin in the population sense caused these neopolyploids to respond differently. And similarly with rhizome allocations, this is the percent of rhizome mass that comprises the total biomass of plant. There's a little bit of nuanced differences going on there, primarily driven by the control and high resource supply levels. So you can see with Cheney, Washington, the only real difference between neotetraploids and their diploid parents was at the low resource supply. But as we increase the resource supply to those plants from Cheney, there was no difference in allocation strategy with rhizome storage. And when we look at, say, Palouse, Washington, or Santa, Idaho, there's different reaction norms playing out based on whether you're diploid or neopolyploid. For Palouse, the neopolyploids and the diploids looked pretty in sync. It's just that the neotetraploids were less apt to store more carbon below ground than, say, like Santa, where there's some opposing reaction norms going on. So population of origin did have a real effect on some of the trait responses for our plants that we looked at. And I do want to just drive this all home and synthesize all of my results by really focusing on this low nutrient treatment, where we saw no statistical difference in growth between the plants. It was only at the high resource supply levels that we saw the neotetraploids were really less efficient at acquiring carbon and getting biomass stored below ground. So these neotetraploids were less efficient at acquiring carbon. They were making stubbier roots. I think it's at these low nutrient supply levels where they might actually have a fighting chance to persist because of that. So I'm just going to use a cartoon example to end on. We can imagine a diploid metapopulation giving rise to many neopolyploid lineages. They respect no geopolitical boundaries for the diploid range. So these neopolyploids are consistently being born through space and time. And then we expect, because of the demographic and physiological challenges of becoming neopolyploid, that it's really in these marginal environments where they might have a fighting chance to persist and ultimately become established. And that's notwithstanding empty niche space filling. So there's been a lot of recent interesting work coming out about mass extinction events in polyploidy. So there's a really big footprint of polyploidy during times of climatic cataclysmic events, such as a meteor strike or a deglaciation event. The neopolyploids of that time seem to have persisted more strongly because they were filling empty niche space. So I really do think that polyploidy is not necessarily a dead end, it's just really environmentally context dependent on when they can become established. So with that, I'll thank my committee and my lab group. Everyone in this photo has really helped me orient myself through grad school and keep me sane most of the time. So I tend to have my head in the clouds with my big research ideas. Everyone's been really helpful in keeping me really focused. And I will say also, the shameless plug, I put my email on the bottom here because I am in the job market for a postdoc opportunity. I am looking to continue my work on studying ecological interactions with polyploids. So with that, I'll open the floor to questions. Oops. And thank you for your time.
All right, thank you, Thomas, for a wonderful talk. As we've done the last few times, please post your questions in the chat, and we'll give you more than a few minutes here to address those questions before we start with the next talk.
Yeah, I'll sort of go through the chain of command and just go through like who goes first. So I'll work from the top and go to the bottom. Give myself a moment to read. My eyes are not what they used to be. How often would neos need to be forming naturally to get a successful neopolyploid establishment? Any evidence that neopolyploid or nitrous oxide treatment affects or limits traits of neos? So we did test the effect of nitrous oxide induction of neopolyploidy, and we didn't see any real statistical difference in any of the traits. There was a marginal effect on total biomass production, but I'll say, I've been saying gas diploids in my head, but the treated diploids that failed to become neopolyploid, the difference in total biomass production, I think, was driven more by their genetic origin, more so than the fact that they became neopolyploid, because we're testing diploids and their direct neopolyploid descendants. But the untreated diploids were not the direct descendants of those same diploid plants. So there is a little bit of a red herring there for understanding that. They kind of get at the idea of how often would neos need to be forming naturally to get a successful neopolyploid establishment. I'll kind of I'll divert and just rely on Ramsey and Chemsky's publication from the early 2000s and say it really is probably based on the gametic non-reduction of chromosomes during meiosis. So I don't necessarily have direct data to support that for Heuchera, but for most plants, I think it's driven more by how often non-reduced gametes are forming in a plant species. So let's see. The next question. Any information on the impacts of neopolyploidy on reproductive traits? Yeah, so a recent graduate from my lab, Laura Porturis, actually did a meta-analysis on neopolyploidy and floral traits. And I think there's a lot of other literature on that. For Heuchera specifically, I see a reduced flower output or reduced production of flowers. I don't necessarily know about seed weights. So that oftentimes, at least what I see for my other system that I work on in Arabidopsis, there's a trade-off between the number or the reproductive output of plants, so the fecundity and the total weight of seeds. So the lifetime fitness of plants can be really complex, but I've seen that with increasing ploidy, yes, there's diminished flower production and ostensibly fewer seeds, but those seeds are heavier and maybe more able to survive in resource-limited environments. So I don't necessarily know. The story hasn't been completed for this species. Let's see, the next question. So do you think that polyploidy, I like that PP, will be better competitors under low resources than high, or are they just stress tolerators? So I think that in the low resource environments, the really key thing about competition, thinking about what David Tillman would say, if you're allocating more biomass above ground, you're going to be able to outcompete a lot of your nearest neighbors because you're forming bigger leaves that are going to shade your competitors. So I think the neopolyploids, I didn't test competition, so these are pot cultures, but I really do think that these neopolyploids would outcompete some of these diploids in these resource limited environments, because while the diploids are concerned more with storing carbon in roots, the neopolyploids were more concerned, I guess, to anthropomorphize the situation, with forming leaves that would outshade their neighbors. So I think there would be a little bit of a feedback there on that. I don't know, so this is very hand wavy of me. So, but that's probably what I say. And are they just stress tolerators? So yeah, it's a slow growing perennial plant. So a stress tolerant plant, it's still responsive to nutrients as we see. It's just not like a competitive plant where it's like, it's only responding to nutrients and all other environmental stimuli will cause it to go, I wouldn't say extinct, but it'll cause it to die. So survival and stress tolerators is not something that many people measure. I didn't measure survival because hardly any of these plants died. So they can really take a beating. So yeah, they are stress tolerators. Let's see. Are these less heterozygous than a new auto in nature, especially if formed via crossing triploid bridge? So I've tried to get diploids and these established polyploids of Heuchera cylindrica to cross and form triploids in the greenhouse, and I haven't had any luck. So I don't know if they would ever form triploids. I don't have data to support that. I think there's been a single case where people have looked in, I think, Heuchera gracilarifolia, it might have been cylindrica, which is what I am reporting on here, but there's been no mixed ploidy cytotype populations that have occurred. So it seems to me that these cytotypes aren't really readily crossing, but I don't know. I guess they could be more heterozygous, not only because tetraploids are forming recurrently through time, but also there might be, as you point out with the triploid bridge, some introgression going on between the triploids and the established polyploids in the field. So That would be really something that would be interesting to look at. I don't necessarily have data to get at that, whether they're less or more heterozygous. The neopolyploids that I used, nitrous oxide was applied on the first division of the zygote. So this is purely whole genome duplication to a T. There's no crossing going on. So at most we have 2 alleles per locus because of the nature of application. Or application of nitrous. Are there any differences in heterozygosity between established tetraploid individuals and the neos? So that I do not know. The neopolyploids, I mean, we've at most got 12 maternal lineages of neopolyploidy distributed from 4 sites. And our established tetraploids that we used, I incorporated only 2 sites from 4 lineages, just as an interesting aside. I wish in hindsight that I'd incorporated more established polyploids to kind of get a better handle on genetic diversity and how that affects the trait responses of plants. But I just didn't have that in the context of greenhouse space and time. So that would be something to look at in the future. Let's see. How old are the established tetraploids? So the established tetraploids, I'm relying on a publication from 2013 from, I think, Kelsey Glennon and I can't remember his first name, but Godso, where they estimated that polyploidy had occurred sometime during the last deglaciation event. So the Pleistocene deglaciation event in the Pacific Northwest. That's where we see the first footprint of Neopolyploidy really playing out in this species. So about 10,000 years, give or take. So I don't necessarily want to get into like what constitutes old versus new Neopolyploids or Paleopolyploid, because I don't necessarily know, but 10,000 years is the short answer to that. Let's see. Many soils is where new polyploids are arise. Can you comment on that? So I think there's a little bit of discussion. I think Donald Levin has talked a little bit about this, and there's a number of other research groups that are currently looking at where neopolyploidy would occur more frequently, and oftentimes they state neopolyploid events or the gametic non-reduction of chromosomes occurs with stress, ecologically speaking. So I would say that in low nutrient environments where plants are ostensibly a little more stressed, they might be forming more non-reduced gametes. So it also might be a consequence the establishment might be a consequence of more non-reduced gametes being formed in those types of conditions. So the context dependency, environmentally speaking, for polyploid establishment might be even more nuanced than what I was suggesting in my discussion. So I think that's a really interesting idea, and I would like to test that at some point, but I haven't necessarily done that yet.
All right, well, let's have you take one more question while you transfer the screen over to Lee so he can get started so we can get that going for the next one. If you could just stop screen sharing and we'll get Lee to share his screen. And then you could take the next question there, and then we'll have to let the rest of those go to after both talks are finished, just to keep on time. But all right.
I'll answer the next question.
Yep.
Email.
Oh, okay. Well, we can pick those up too afterwards. I think we're at Jeanette Witten's question there. All right, just to keep that in mind. All right. Well, thank you, Thomas, for a wonderful talk. And everyone, we have plenty of time afterwards to finish answering all these really interesting questions, because I actually want to hear the answer to these questions myself. So I hope Thomas will stick around and be able to do that for us. All right. Thank you, Thomas. All right, I'll go ahead and get started with our next speaker today. Is Zheng Li. Li did his undergraduate career at Miami University in Oxford, Ohio. He's currently a PhD student, actually here in my lab at the College of Evolutionary Biology at the University of Arizona. Li is graduating soon, and we'll be starting a postdoc sometime here in the fall, but when he's ready to tell everyone where that's at, he'll let us know. Today, Lee is going to tell us about research that he's been conducting for the last few years here, studying ancient polyploidy, and in particular ancient polyploidy and a low rate of chromosome loss explain ferns with high chromosome numbers. With that, I'll let Lee take over.
All right, thank you, Mike, and thanks everyone for coming. So as botanists, biologists, we're all interested in variations in species. So in here we have all this flying plant and fern. What I found that's really interesting and cool is if you do a chromosome squash on all those guys and all the flying plant in here, including grasses, asters, has n equals 2, including this xanthosma, which our lab is going to sequence the whole genome. And compared to the Ophioglossum reticulatum, which has n, the haploid chromosome count more than 1,000. And of course, this guy's Ophioglossum reticulatum is an outlier, has n equals 1,260, which has the highest chromosome count among all the eukaryotes. And the chromosome number variation has been studied. People know that among major lineages, in here we include fern, lycophyte, and eudicot. Among those major lineages, there are lots of chromosome number variation exist. So for example, in the figure that we're showing you here, on the x-axis is showing you the haploid chromosome count, and on the y-axis is showing you numbers of taxa. So first to look at is among all those 3 lineages, look at a range of the variation. So in ferns, of course, Ophioglossum is not included in here, but the range goes from almost 0, pretty low, to all the way to more than 150. And also in lycophyte, the range is also pretty high, from n equals 9 all the way to more than 200. But in eudicot, in here, we see the range is a little bit less compared to fern and lycophyte. And the other thing to look at is the common number for each lineages. So in fern, the most common number is 41, and also other common numbers like 36, 72, 82, some of this showing you patterns of polyploidy. So those are the common numbers. And the most common number in lycophyte is n equals 9, followed by 22, and then 34. But in eudicot, what we're seeing is the most common number is n equals 9. And one of the different patterns for eudicot compared to other lineages is we're seeing lots of different spikes here, suggest there's lots of aneuploidy or disaploidy. Those are individual gain and loss of a single chromosome going on within eudicot. But we don't see that in ferns and lycophyte. And sort of the study for haploid chromosome count has been going on for a really long time. So one of the classic study is from Kalkowski and Baker in 1966. So what they found is the average haploid chromosome count among these 3 groups. In Homospherous fern, it has a haploid chromosome count for 57, which is really high, 3 times compared to flying plant, compared to heterosporous ferns. So how do we explain this really striking pattern? Well, there's multiple hypotheses has been proposed to explain this pattern. So the first hypothesis is the ancestral high chromosome number, first proposed by Duncan and Smith in 1978. So what they suggest is, well, fern may have just ancestral high chromosome number. But from our chromosome analyses, using chromosome, looking at chromosome number evolution in ferns, what we're seeing suggests it's not very likely. So in here, this figure is showing you the x-axis, just showing you the distance from the root towards the tip. And on the y is showing you the inferred ancestral chromosome number. So if you're moving up from the root towards the tip, we're actually seeing increases of chromosome number rather than ancestral high chromosome number. And the other hypothesis that has been proposed is the ascending aneuploidy or disaploidy. And in flying plant, we know that there's lots of aneuploidy or disaploidy going on. If that's the case, this is what we expect to observe in this kind of figure. But from Otto and Whitten 2000, we're seeing in fern, well, that's not the case. We don't see that many spikes in fern, suggest there's less amount of aneuploidy or disaploidy going on. So this leaves us to the leading hypothesis, the multiple round of ancient polyploidy or ancient whole genome duplication in ferns. So this idea first proposed by Hoffler and Sodas in 1986. So they hypothesized that fern may have experienced multiple rounds of ancient polyploid speciation, followed by gene silencing, but not chromosome loss. And Barker and Wolf in 2010, they further compared that to flying plant and further asked the question, so why do homosperous fern have so many more chromosomes compared to flying plant? Especially in flying plant, we know that there's lots of ancient polyploidy going on. From our most recent study from the 1KP 1000 plant transcriptome project, we infer 173 ancient whole genome duplication event across the flying plant phylogeny, and we estimate about 75% of the flying plant family has ancient polyploid origin. So this go back to the question, so why do homosperous fern have so many more chromosomes compared to fern? Why do homosperous fern have so many more chromosomes compared to flying plant? So the first hypothesis, homosperous fern have more round of ancient polyploidy. And the other hypothesis that we have is, well, maybe homosperous fern have similar or even fewer round of ancient polyploidy, but they tend to not lose their chromosome, or they're losing their chromosome in a really slow process. So to test those hypotheses, what we first need to address is where in the fern phylogeny did this ancient whole genome duplication event occur, and what are the frequencies of ancient polyploidy in both ferns and flying plant. So to address those questions, we assemble a data set, probably the largest data set for ferns genomics, including 142 fern genomes and transcriptomes, a large chunk of data coming from our 1KP analyses, and then also the Shen et al. 2018 paper also have quite a few of new fern transcriptome that's available. So how do we infer this ancient whole genome duplication event? There are three major approaches that people has been using in genomic studies. So number one is the age distribution of gene duplications, also known as ks-plot. The data required for this type of analysis usually is single transcriptomes or genomes. So you run a ks-plot for each transcriptomes or genomes. So in this type of analysis, we look at peaks that are result from the whole genome duplication event. And if a species doesn't have experienced the whole genome duplication event, we do not expect to observe those peaks in the histograms. And the other common type of approach is the tree-based approaches. There's many methods have been developed for this type of approach. The data that required usually is multiple transcriptomes or genomes. In general, a cluster gene family phylogenies and building gene trees. And you look at patterns of gene duplications based on trees or subtrees across the phylogeny to infer ancient polyploidy. And the last one is the syntenic analyses, which require high-quality genome assemblies. And synteny just means the collinear blocks in the genomes that result from the whole genome duplication event. And this is usually considered as the gold standard for inferring ancient genome duplication event. So how do we infer those in ferns? Well, we take all those three different approaches with the data set that we have. For ks-plot, we use our DuPipe pipeline, which is available in the evopipe.net. And for the tree-based approach, we use the multi-taxon paleopolyploidy search, or MAPS, that I have been developed over the last couple of years. And also with the first two fern genomes, Azolla and Salvinia, and we also using the Koji platform to look at syntenies for those genomes. So we run the analyses on all the data that we gather, and of course I'm not going to show you every single one of them. So I'll just go through examples for each type of analyses. So for DuPipe, for ks-plot, this is what we have. So for example, in the acquisitive transcriptomes, we do observe peaks associated with whole genome duplication event. And in Moratia, we also seeing a whole genome duplication event that we had. And also in tree ferns, we see 2 rounds of whole genome duplication event. And also in pteridium, we also see a peak associated with the whole genome duplication event. So with the tree-based approach, I want to show you one example from the Osmundaceae. So in here, we found that there's 2 Osmunda species and also one of Osmundastrums. All those 3 guys share the Osmundaceae whole genome duplication event. And also in the Saxifragaceae, the tree ferns, we see some species have experienced 2 rounds of whole genome duplication event that share among the Saxifragaceae. And also with the syntactic analyses, we have the first 2 fern genomes, Azolla and Salvinia, in collaboration with Zheng Li at Cornell University. So with the high quality genome, we first look at the syntonies from Koji, so getting those colonial blocks in the genomes. And what we do is we extract the duplicate genes from those regions, and that's called the syntalogs. And we first look at the divergence between Azolla and Salvinia in terms of KS, the synonymous substitution per synonymous site. So here's the divergence represented with blue in here. And what we see is we see one whole genome duplication event, the correlates whole genome duplication event that predate the divergence of Azolla and Salvinia. And we also seeing there's one duplication event, the Azolla specific whole genome duplication that happens after the divergence of Azolla and Salvinia. And with the genomes and transcriptomes, we also run the tree-based approach in here. And you can see that the 2 results are highly consistent. So with our maps analyses, the blue line represent the actual result, and the multiple gray line represent our null simulations. And what we found is here, the blue dot represent the divergence of Azolla and Salvinia. And we also, in here, from the maps analyses, we've seen one duplication event associated with the Azolla, and then there's another whole genome duplication event shared by the cornlaptosporangia, but not shared with Lygodium. If you look at the color, the result is consistent between these 2 different analyses. So across our data set for 142 fern genomes and transcriptomes, we inferred 26 ancient whole genome duplication events during the evolutionary history of ferns. And going back to our question, so why do homospores ferns have so many more chromosomes than flying plants? Well, hypothesis number 1, we have fern has more round of ancient polyploidy compared to flying plant. And hypothesis number 2, maybe they have similar or even lower rounds in fern compared to flying plant. So to test that, we need to estimate the round of ancient polyploidy in both fern and angiosperms. So how do we do that? So here I give you a toy example that we have. This is a phylogeny, and each of the species, the red dot represent the inferred ancient whole genome duplication event. So for each species, we count how many rounds of ancient polyploidy that each species experience. So for example, with species E, we'll count 1, 2, 3 rounds. And for example, for species D, it will be from the root towards the tip, it will be only 1 round. So this is what we have for the ferns. We do that for each species that we have in here. And for other lineages, other vascular plants, we use our previous estimate from the 1KP project. So this is what we got. So for ferns, on average, each fern species has experienced about 4 rounds of ancient whole genome duplication event. Some species can go up to 5. Some species go down to 2, but average is around 4. And in flying plant, we also seeing that the numbers of round of ancient polyploidy on average is about 4. Some goes up to 6, and those are mainly in the Brassicaceae, and the one that go down to 2 is the Amberilla. So if you put that the fern and angiosperm together, you see the pattern is actually quite similar. And we also including other estimate from gymnosperm and lycophyte, and we compare the distribution. So there's no significant difference between angiosperm and fern, but there is significant differences between fern and gymnosperm and fern to lycophyte. So also, we need to keep in mind that fern is a really old lineages. The crown group age go back to probably more than 350 million years ago. So given the lineage that is old, we want to take the lineage time into account. So we further divided the crown group age to estimate the rate of polyploidy. So this is what we have. So in here showing you flying plant definitely has the highest rate of ancient polyploidy per 1 million years, followed by fern and then gymnosperms. So here just to give you a comparison between the round of polyploidy and the rate. So going back to our two hypotheses, either fern has more round or similar rounds, well, our results suggest fern and angiosperm probably have a very similar round of ancient polyploidy, supporting our hypothesis number 2. And also, this rate estimate rate of ancient polyploidy is coming from our phylogenomic analyses, so completely based on inferences from the transcriptomes and genomes, and then estimate the rate. We also have a completely different method and data set to do the phylogenetic reconstructions of chromosome number using the Chromovol tool. So this, in Chromovol, it takes in the chromosome count and also the phylogeny. So we have the phylogeny for angiosperm, fern, and gymnosperm, and we also have pull out the chromosome count from the databases. So we estimate the rate of polyploidy per 1 million years, and we see the overall pattern is really similar to what we found before. Angiosperm flying plant definitely have the highest rate of polyploidy compared to ferns, and gymnosperm, fern is the second, and then gymnosperm has the slowest rate. And if we put this together, the phylogenomic analyses and the phylogenetic reconstruction of chromosome numbers, we're seeing the overall pattern is consistent between these two different analyses. And also, if you look at the actual number, it's actually really similar. So we estimate in angiosperm, it's about 0.02 event per million years. And then here it's 0.014, so it's pretty close. And also in ferns, about 0.01 in both of the analyses. And we also using the Chromovol analysis to estimate the rate of ascending disciploidy and aneuploidy. So in the red color here represent the flying plant, and the blue, the green is the fern. So we can see that there's lots of disciploidy going on in flying plant. The rate is much more higher compared to ferns. So I think we have the really comprehensive evidence to show you ancient polyploidy and low rate of chromosome loss explain fern with high chromosome numbers. So to summarize our result, where in the fern phylogeny did this ancient whole genome duplication event occur? So I've shown you the phylogeny. Overall, across the fern phylogeny, we have 26 ancient whole genome duplication events. We also show you the synthetic evidence, phylogenomic evidence of ancient whole genome duplication in the first two fern genomes, the Azolla and Salvinia. And go to our question. So why do homosphorous fern have so many more chromosomes than flying plant? Well, what we found supporting the multiple ancient polyploidy hypothesis first proposed by Hoff and Sotis a long time ago. And what we observe is homosphorous fern have similar round of ancient polyploidy and a lower rate of chromosome loss. So with that, I will thank everybody from the Barker lab, especially to Shane that has been putting the Chromovol analyses, and also everyone else has been helping me a lot with the analyses. And also to my friend Christian and my friend from China, Yifan Li, who give us great illustration of those ferns that you see on the phylogeny, and also EEB department at UA and our funding agency. With that, I'll take any question that you have.
All right, well, thank you, Lee. We'll let everybody type their questions in the box here, and give him a few rounds of questions, and then we can go back to the other questions as well for Thomas to get those going. And we don't have a time limit on the end of this, so I'll let folks stick around as long as we need to. I'll keep recording as Lee answers the questions, and then once we get through all the question answering from both Lee and Thomas, we'll turn that off. And as always, we'll post these recordings later on this afternoon so people can check it out. With that, I'll let Lee take over the question answering right now.
Cool. Okay, trying to see which question is the first.
Looks like it's from Jonathan.
Okay, got it. So what is it about fern compared to flying plant, chromosome, or reproductive biology that can explain the low rate of chromosomenizations in fern? I think we have some ideas, but not a whole lot. I think definitely could be something going on with the swimming sperm, and also what strategy they have for chromosome pairing and meiosis. And we have some analyses that's coming up. So I don't think we have a full answer right now. Some of the mechanisms need to be figured out, but stay tuned. And then also just to advertise, we're gonna have the bioarchive released pretty soon for our analysis. So there might be a little bit more detail in there that you can look at. So from Rob, I think the question is really similar. Is there something about fern reproductions or chromosome that might contribute to the retention of the chromosome over time? Yes, for sure. The detailed mechanism is largely unknown. So the chromosome in fern is really interesting. So they have highly uniform chromosomes, different than flying plants. So you actually really hard to tell them apart when you're doing the squash. They all looks kind of similar. Well, there could be many things that contribute that. The other thing that I've been thinking about, it could be the sort of the methylation pattern, but actually very few people have been looking at that. So I think the detailed mechanism is just largely unknown. There's probably many things that will contribute to that. So have ferns compromised their lifestyle in some form due to their large genome size? Not really. I'm not really sure about that. They definitely have pretty different lifestyle compared to flying plants or other lineages, but not sure that really have anything to do with their larger genome size. They seem to doing fine, seem to doing their own thing, but not sure whether that really due to their large genome size or not in terms of their lifestyles. So the next from Justin, are there types of chromosome mutations, inversions, translocations as rare as chromosome fusions in ferns? If not, what makes the fusions different? As rare as chromosome fusions, so we haven't looked at the inversions and translocations, which it would be really great. I think what we need is actually one genomes from the homosporous fern, because the current two genomes, Azolla and Salvinia, both heterosporous ferns. It would be really great if we have the homosporous genome so that we can look at these inversions and translocations in those genomes as well to compare. And what makes the fusion difference, I don't really know. And I also don't think anyone really know what mechanism that's driving that. We know that they just have highly uniform chromosomes, seem to have a pretty big chromosome. I don't know, maybe Mike has any opinions on that, but I don't think anyone has a really sort of deep understanding on what exact mechanism that's driving that. Yes, so why heterosporous ferns seem to follow the pattern of genome size, genome evolution that we see in angiosperm? Great questions. We also highly interested in that. And also thank you for bringing up the heterosporous lycophyte versus homosporous ones. So that's the next things that we really want to look at. And we thought about maybe that's sort of related to the swimming sperm that they had with the heterosporous organisms, but the detail seems not clear. It could have something associated with that. I personally think that it might associate with the genes that's driving chromosome pairing behavior during meiosis, and that's sort of what we sort of keep continuing looking at.
All right, well, I think that might be, oh, okay, I'll let Justin there ask the last one. There's one more question there, Lee, from Justin, and then we'll let Thomas go back and answer some of those other questions that were left over from his, and then we can open up discussion. So Lee, if you want to take that last question there from Justin Conover.
Okay, from Justin. So do you have any idea of the frequencies of allo versus auto in this? So I don't, because most of our estimate from the ancient polyploidy has been pretty old, and we don't really have a good estimate for which one is allo, which one's auto. I wish our next step, once we develop some other tools that can help us to infer that, but I think the frequency probably relies on, if we really want to estimate the frequency, probably we will be relying on looking deeper into the recent polyploidy that's happening in ferns.
All right, well, let Thomas go back up here. If you want to hop back on the microphone there, Thomas, and I can help field these questions. You got a couple of questions left there.
Yeah.
And then we'll open it back up and everybody can just kind of chat for a little while. We can maybe figure out some of these problems. So back up there to Jeanette's question, Thomas, do you have a sense of whether the differences in performance that you see under different nutrient conditions are large enough for neos to persist long enough to establish and become locally adapted?
Yeah, so I've got the remaining questions brought up. I don't necessarily know that I have that because I only have like a single generation of Neopolyploids and across just one growing season. So for a perennial plant, tracking the lifetime fitness is really complicated here. It would be really great to do a long-term study looking at how Neopolyploid seeds that are formed each season kind of build up their population or go extinct. But I think that would take a lot of doing. I really do think it's in these low resource environments where the odds of Neopolyploid establishment, although not great, they're at least there. And they're a high nutrient environment. It appears that 100% of the time, the diploids are going to outpace their Neopolyploid progeny in performance. So I don't know, I guess is the short answer to that. Really interesting. And then Chris Pyers had another question. Is there anecdotal evidence about whether soil types differ where neopolyploids have survived in the system. So I'll take that where neopolyploids have survived to mean the established tetraploids we see out in the field. So in 2018, when I collected my seed stocks of established tetraploids and diploids, I had a few extra field sites that I didn't incorporate into this study, but I always collected about 1 kilogram of soil from each site and looked at a bunch of different physical characteristics. Like I looked at the pH, the electrical conductivity, which gives you an idea of how much mineral nutrients are floating around in the soil horizon. We looked at like plant available nitrogen and phosphorus from the field. We didn't really see any appreciable differences, but this is mostly working in the parapatric zone, or like where the diploid and the established tetraploid ranges abut one another. I didn't go as far north as like the middle of British Columbia, where the established tetraploids also reach in their range, or I didn't go to southern or middle of California where the diploids go to. So there is a little bit of a geographical range difference going on where the diploids and established tetraploids don't co-occur, but we need more data to really say what the general physical characteristics of soil types that the established tetraploids versus the diploids might prefer. Let's see, Hirsch Green, we found the neopolyploids were intermediate to diploids and tetraploids in fireweed. Others more like tetraploid. Curious what generation these neopolyploids were. So right, these were first generation neopolyploids. So what I did when I put the flowering diploid that I had self-pollinated into the nitrous oxide chamber before pressurizing it, we always like tracked which flower had been pollinated and then collected seeds from that, and then just took those seeds directly after they had formed and matured and sowed those directly into the next experiment. So we actually had to use flow cytometry to verify that the final cytotype of the plant was indeed neopolyploid at the end of the experiment. And that's kind of what lended its way to allowing us to look at whether something was a failed induction diploid, which we had planned comparisons of for the beginning, but we had more statistical power for because we had some migrants that we had at the very beginning or the onset of the experiment classified as putative neopolyploids, but after flow cytometry, it became known that they were failed induction diploids. So these were first generation polyploids in the very literal sense. Let's see. And then I think I had one more question. How did you check that the neopolyploids were not really variable among themselves in their traits? Right, so I always looked at population of origin. We didn't, because of the nature of having to verify the final ploidy or cytotype of the plant via flow, we didn't have full nutrient treatments within lineage. So we had 12 neopolyploid lineages. But because some of them turned out to be failed induction diploids, we weren't able to fully track the lineage specific effects. So that's why I grouped by population of origin rather than independent lineage. So that was kind of a sad outcome, but I made lemonade from those lemons, as it would be said. And we didn't really see a whole lot of variation in the trait responses amongst those neos within their population of origin. And that kind of came out with our standard errors or our 95% confidence intervals in our statistical models. I think we had some pretty good precision of our estimates. Some traits are more variable than others, of course. The biomass being one of the least variable, nutrient concentrations being the most. That might be an artifact of the machine we measured on because of measurement error from the machine. I didn't use the most sophisticated tools to measure those traits just because I'm poor. They were not that variable. I think that was the end of the questions that I had from the chat. Let's see. Oh, there might be more recent questions. Oh, nope.
All right, I think that wraps up those questions. I want to thank both Thomas and Lee for today's talks. Those were great. I just want to put a plug in for the speakers in 2 weeks. We have Paulina Tomaszewska, who will speak on cytogenetics and polyploidy to exploit biodiversity in Euroclea, tropical forage grasses. And Lex Flegel, who will tell us about solving hard population genetic problems and polyploids using deep learning. So it should be a fun couple of talks that again span the diversity of approaches and questions that we ask in polyploid species. And with that, I'm going to go ahead and shut down today's recording.